آسفة على العطل اللي صار. يعني علق الكمبيوتر عندي. أنا دكتورة إجلال أبو الرب. اليوم رح أعطيكم الفسيولوجي بارت للاندوكراين سيستم. هلا أي سؤال عن المحاضرة إذا ممكن نخليها للفلكسبل لكشر اليوم الخميس، أو إذا أي حدا ممكن يبعث لي الكويشنز تاعونه على الإيميل تبعي. رح أبعث لكم إياه إن شاء الله مع الدكتورة فاطمة. هلا رح نبلش المقدمة للاندوكراين سيستم. هلا انا شايفه كل الطلاب نبلش ولا نستنى ليجوا طلاب اكثر؟ يعني معلش اذا بتحكوا لي يعني. هلا رح نبلش introduction على الاندوكراين سيستم. Before we start introducing the endocrine system, we want to talk about an important concept for various biological processes in our human body. Now, in order to achieve our different physiological and biological processes in our human body, the cells need to communicate with each other. Now, the ability of cells to communicate with each other is crucial to control many processes in our body. Now, the cells can communicate with each other by using different type of chemical messengers. We have for example, first of all, the neurotransmitters which are secreted by the axon terminals of neurons into the synaptic junctions, and these neurotransmitters, they are used to control the function of other nerve cells. Now, the neuroendocrine hormones, which are specific type of hormones secreted by neurons to the blood in order to affect the function of distant cells, like for example, the hypothalamic hormones. The third way the cells communicate with each other is by the using of paracrines. Paracrines are messengers or chemicals secreted by the cells into the extracellular fluid in order to affect the function of a nearby cell. On the other hand, the autocrines, which are chemicals secreted by the cell to affect the function of the same cell that secretes these autocrines. The endocrine hormones, they are chemicals or messengers secreted by specialized cells or glands into circulating blood in order to function on distant cells to achieve certain or specific physiological responses. Now, this schematic diagram represented different types of chemical messengers the cells used to communicate with each other. Now, our system is the endocrine system. We will talk about the endocrine way of coordinating the function of different types of cells, tissues, and organs in our body. So the endocrine system is a collection of specialized cells that form specialized glands. These glands, they are secreted chemicals or ligands called hormone, which will regulate the homeostasis of in our body. Now, These hormones, they are important to regulate the growth and the reproduction function of our body. They regulate the balance of the water, electrolytes, and the nutrients in the blood. Also, they are necessary. They are necessary to regulate the immune system. Also, they regulate the metabolism and the energy balance in our body. Now, this scheme or diagram showed the different endocrine glands we have in our body. So some of these endocrine glands, they are dedicated endocrine glands, means their main primary function is to function as endocrine glands. Other endocrine glands, they are their primary function is not endocrine, but they secrete some hormones to regulate certain processes in our body. And also we have in some organs specialized cells that secrete certain hormones or required for the peripheral conversion of hormones into their active form. So I will mention these glands briefly here, and we'll talk about them in the upcoming lectures. So the first endocrine gland is the hypothalamus, and the hypothalamus we call it the control gland. Now, the hypothalamus has another gland attached to it, which is the pituitary gland, and the pituitary gland is the master gland in the endocrine system. And we have the pineal gland. In the neck, we have thyroid glands. And behind the thyroid glands, we have the parathyroid glands. Also, we have the adrenal glands, which are located on the top of the kidneys. And we have the endocrine part of the pancreas. And we have the gonads, which are the ovaries in female and the testes in males. And also, as I mentioned, there are some organs who have specialized cells that secrete some hormones or helps in the conversion of inactive hormone to its active form. These tables summarizes the hormones secreted by different glands in our endocrine system. And this table shows that some hormones that required peripheral conversion in order to become active and exert their function. Yes رزان.
دكتور نحفظهم كلهم أو كيف بدي أسأل عليهم؟
هلا يعني هم يعني هلا يعني بنحكيهم احنا بريفلي، بس لازم نحفظهم لانه بالمحاضرات القادمه رح نحكي عنهم كلهم عن الوظائف تبعيتهم. هلا الاندوكراين glands, why we call them endocrine? These glands are ductless, so they secrete the hormones directly to the blood, and the blood will transport these hormones to distant target cells. Now, these target cells, they can't respond to this hormone unless if they have specialized receptors for that to that hormone. Now, the endocrine system needs regulation, and the predominant mode to regulate the endocrine system is the negative feedback. Negative feedback, briefly, when substance A, when it stimulates the secretion of substance B, and on the other side, substance B will stop the secretion of substance A. This is simply the negative feedback. Now, why we need negative feedback in our endocrine system? Negative feedback is important to to ensure that the hormone range to ensure narrow hormonal levels. Means I don't want too much hormone, and I don't want too less of that hormone. So by this we are stabilizing the level of hormone to meet the required physiological processes. Now, in a few circumstances, we have positive feedback or control to the endocrine system, and this occurs in certain processes where we need a gut to give processes like, for example, during ovulation or during the delivery of the fetus. Here we need to have a high level, continuous high level of certain hormones to achieve the process, and after that the level of hormone will decrease. So this positive feedback only we need it in specific or emergent situations. Now, we have two types of negative feedback loops. We have the response-driven feedback loop, and we have the endocrine axis-driven feedback loop. Now, in the response-driven feedback loop, now the target or the physiological response, or we can say the end result or the outcome of the hormonal action controls the function of the endocrine system. So, for example, when we have high glucose levels, this will stimulate the secretion of insulin, and in the livers, this will stop the hepatic gluconeogenesis, and in the muscle, we increase the uptake of glucose by the muscles. So this is what we call response-driven feedback loop. Here, the main regulator or inhibition, stimulation or inhibition of the endocrine system is by the extracellular parameter other than hormone. In the endocrine axis-driven feedback loop, here the peripherally produced hormone is controlling the function of higher endocrine glands, the hypothalamus and the pituitary gland. The endocrine axis-driven feedback loop, the endocrine axis feedback loop it differs from the response-driven feedback loop by the fact that the physiological response or the extracellular parameter other than hormones play a minor role in the regulation of, we can say, the higher glands or the master glands. Now, also there is another important regulation system for the releasing of hormone. We have a specialized collection of neurons or region in the hypothalamus called the suprachiasmatic nucleus. The suprachiasmatic neurons, they are responsible of creating what we call it the circadian rhythm. The circadian rhythm play a role, an integral role even, in controlling the releasing of various hormones in our body. Now, the suprachiasmatic neurons, they impose what we call it a 24 to 25-hour clock, which is in synchrony with the normal environmental light-dark cycle created by the Earth's rotation. Now, this suprachiasmatic nucleus in the hypothalamus is regulated or is stimulated by two ways, either by specialized light-sensitive retinal cells, which have photoreceptors that send signals to the suprachiasmatic nucleus via the retinohypothalamic tract. And the second regulatory or stimulating system for the suprachiasmatic nucleus is the pineal gland. The pineal gland connected the various physiological processes to the with the suprachiasmatic nucleus. Now, this pineal gland, which is close to the hypothalamus, it synthesizes the hormone melatonin. Melatonin, the rate-limiting enzyme in the synthesizing melatonin is the N-acetyltransferase. The N-acetyltransferase levels, they oscillated in a cyclic fashion. So, which will regulate the levels of melatonin, and as a result, we are regulating the other hormones. Now, the synthesis of melatonin is peaked in the dark, while it is inhibited in the morning or in the light. Now, melatonin will transmit a message or signal to the suprachiasmatic nucleus that the night time has arrived. So as a result, the body functions as well as the hormone release will be regulated accordingly. So the melatonin feedback to the suprachiasmatic nucleus is important to regulate the day-night clock or the circadian rhythm. This figures or this diagram is an example of how the circadian rhythm controls various biological functions and hormone release. For example, the growth hormones, they peaked in the midnight while they decreased in the morning. On the other hand, the plasma adrenocorticotropic hormone, it decreased in the midnight while it increased in the morning. This also figure shows also how the circadian rhythm control different hormones in our body. For example, we have the sleep hormones like the prolactin, the leptin, and the ghrelin. On the other hand, we have morning hormones which peak in the morning, like the cortisol and testosterone. The circadian rhythm, or when we understand how the circadian rhythm controls the hormone function, we can understand how the fluctuation in our biological function. And also it is very important when we are designing certain drugs or certain molecules to treat different conditions. Now, what are the biochemical classifications of hormone? We have protein or polypeptides, which comprises the most hormones in our body. Now, the size of these hormones, they are ranging from small peptides with 3 amino acids like the thiotropium releasing hormone, and we have large proteins with 200 amino acids like the growth hormones and the prolactin. Now, these hormones are synthesized in the polyribosome. So the synthesis of proteins start with the nucleus when we are releasing the primary gene transcript, which need modifications like by the excision of introns, the splicing, the capping to produce the mature messenger RNA that will enter the cytoplasm. In the cytoplasm, the polyribosome will translate this mRNA into the pre-pro hormones. The pre-pro hormones has a specific N-terminal 15 to 30 peptides called the signal peptide. This pre-pro hormone will be transported to the rough endoplasmic reticulum, and in the rough endoplasmic reticulum, it will be the N-terminal signal will be cleaved, and to generate the pro-hormone. The pro-hormone then will be processed and packaged in the Golgi apparatus and stored as active hormone, which will be secreted by oxocytosis. The oxocytosis of protein or polypeptide hormones, it is a regulated process. Means these hormones or peptide hormones, they are not secreted continuously. They are secreted when in what we call a stimulus secretion coupling process. Means when we have a stimulus, like for example intercellular calcium, the secretion of these hormones by oxocytosis will be initiated. Otherwise, they will stay stored in the specialized vesicles. These hormones are hydrophilic, so they can't cross the cell membrane. And they will act on a surface or extracellular receptors, and they are circulating in the blood in an inbound form, and therefore they have a very short half-life, and these peptides can be removed from the blood by endocytosis or lysosomal degradation. Now, many of these peptide hormones are small in size, and they are secreted in its physiologically active form, like for example the luteinizing hormones and the placental human chorionic gonadotropin, HCG hormone. And these hormones, they are useful when we diagnose certain condition. For example, when we need to detect the pregnancy, we are using the HCG hormone in the urine. The peptide hormones, they are digested extensively in the GI system if administered orally. The second type of hormones, we have the steroid hormones. The steroid hormones, they are produced in the adrenal cortex, ovaries, testes, and placenta. And these hormones, they are synthesized by a series of enzymatic modification of cholesterol. So the precursor is the cholesterol in the synthesis of steroid hormones. These hormones are hydrophobic, so they can cross the cell membrane easily, and they act on an intracellular receptors to enhance the expression of various genes. The steroid hormones, they show some degree of specificity toward receptors. So sometimes we have, we can say, connected function of different steroid hormones, and their effect lasts for longer than the peptide or protein hormones. So because of their hydrophobic nature, these hormones don't need to be stored, and they can be secreted when the cells need or when there is a need to these hormones. Also, these hormones undergoes extensive peripheral conversion to in order to release the active form of these hormones, like when we have the conversion of cortisol to cortisone in the kidney. And sometimes disturbances in this peripheral conversion of the steroid hormones can lead to various endocrine disorders. Also, these hormones, because they are hydrophobic, as we said, they are transported in the blood by binding to the non-specific proteins or transport proteins like albumins, or a specific type of transport proteins, globulins. And these hormones, because they are water insoluble, in order to secrete them in the urine, we need to inactivate these hormones by the liver, like for example, sulfur conjugation in the liver, to increase their water solubility and decrease their affinity to bind the transport proteins. So now, after modification, this modification in the liver, these hormones they can be excreted easily in the kidneys. Now, these hormones, different from also peptide hormones, they can absorb easily from the GI system. And this table is an example of these hormones. Like we have the progestin, we have the glucocorticoids, we have the mineral corticoids like the aldosterone, we have the androgens like the testosterone, and the estrogen like the estradiol. We have the third hormones are the amine hormones. The amine hormones, they are tyrosine derivatives hormones, and we have two types of these hormones: the thyroid hormones and the catecholamines, which are the epinephrine and norepinephrine. The thyroid hormones, or we call them the iodothyronines, they are made by coupling iodated tyrosine residues through ether linkage. And these hormones, they are water insoluble, so they are transported or circulated in the blood by binding to specific globulins, like the thyroid hormone binding globulin. These hormones, they have long half-life. For example, the T4, the half-life of T4 is 7 days, and the half-life of T3 is 18 hours. These hormones act like the steroid hormones on intracellular receptors, and they can be absorbed in the GI system. The catecholamines, on the other hand, they are synthesized in the adrenal medulla, and the main hormone synthesized in the adrenal medulla is the epinephrine, with lesser extent the norepinephrine. These hormones, like the peptide hormones, they are hydrophilic, they can't cross the cell membrane, so they produce their action by binding to extracellular membrane receptors. Also, they are water soluble, so they are circulating in the blood as free hormones, or sometimes they loosely bind to the albumin. Like the peptide hormones, these hormones have short biological half-lives, 1 to 2 minutes. Now, as we mentioned, the hormone can't exert its function on the target cell without the presence of a specific receptor on this target cell. So the hormone receptor binding is the crucial step in initiating the function of the hormone. Now, the binding of hormone to its designated receptor induces conformational changes in this receptor, and this conformation change we call it as signal. This signal will activate one or more intracellular pathways or messengers. Now, these messenger molecules will ultimately produce the physiological effect, or activate the effector proteins to induce the specific cellular function needed for that target cell. The combination of hormone receptor binding, the signal, the activation of the messenger, we call the transduction, and the regulation of the effector proteins or the physiological response, we call the signal transduction pathway. So, which will lead to the final outcome, which is the cellular response. Like, for example, the activation of cell proliferation, survival, chromatin modeling, activation of various genes, transcription. This is, we call it the final outcome of the hormone receptor binding pathway. Now, so the loss or inactivation of the hormone receptors lead to what we call it the hormonal resistance, while constitutive activation of certain hormone receptors, they can cause uncontrolled upregulation, and as a result, we will end up having hormone-independent activation of cellular processes, which will sometimes occur in cancer or in certain endocrine diseases. Now, the endocrine diseases can arise from either abnormal in the expression, activity, or combination of both of these hormone receptors. Now, the hormone signal can be terminated by different way, by the internalization, for example, of the hormone receptors, by the dephosphorylation of these hormone receptors, by proteasomal degradation of the hormone receptors, or by the generation of inhibitory messengers or molecules. Now, what are the different types of hormone receptors? We have many types of hormone receptors that are specific based on the function of the target cells. The first type is the ion channel linked receptors, like the receptors where the epinephrine and the norepinephrine binds. And these receptors, they are attached directly to a voltage-gated channels like the sodium, potassium, or chloride channels, and the binding of this hormone to the ion channels receptors causing the opening or the closing of this channel and control the releasing of various ions in the cytoplasm. The second type of hormone receptors is the heterotrimeric GTP binding protein, or we call it the G protein coupled receptors. The G protein coupled receptor is the major type of hormone receptors in our body. The G protein coupled receptors, the G protein here acts as a first messenger, which will send messages to the intracellular secondary messenger molecules. Now, the second messenger will activate other proteins or other enzymes to achieve the final outcome or the physiological effect needed or required. Now, so as we said, in the G coupled protein receptors, the first messenger is the activation of G protein. Now, what the second messenger, there are different types of examples of second messengers that require to activate or achieve the final outcome or the physiological response. We have the adenyl cyclase, cyclic adenosine monophosphate, or adenyl cyclase, cyclic A and B messenger. And this in this pathway, what happens is that when we activate the G protein by the exchange of guanosine triphosphate to guanosine diphosphate, by that the G protein will get activated, and it's causing the dissociation of its subunit. And here the alpha subunit of the G protein will bind internally the adenyl cyclase enzyme. The adenyl cyclase enzyme will activate or induce the formation of cyclic A and B from A to B. And cyclic A and B play a role in the inducing several kinases. The major one is the protein kinase A signaling pathway, which is required for channel opening or for other biological functions in the cells. The second type of secondary messengers in the G-coupled protein receptors is the calcium ions. Sometimes the activation of G protein lead to the opening of calcium ion channels in the mitochondria and endoplasmic reticulum, and the calcium will be released from these organelles and enter the cell after binding to a specific protein called calmodulin, and this calcium calmodulin complex will activate various enzymes and molecules, for example, like myosin light chain kinase, which will control the smooth muscle contraction. The third type of second messengers in the G-coupled protein receptors is the phospholipase C. In this secondary messenger pathway, the activation of G proteins will, G-coupled protein will activate the phospholipase. The phospholipase will cause the breakdown of some phospholipids in the cell membrane, like for example, phosphatidyl inositol biphosphate, BIB2, and this will further activated into inositol triphosphate and the diacyl glycerol. Now, the function of IP3 is to activate the calcium channels in the mitochondria and the plasma reticulum to release and induce the calcium signaling pathway. And the DAG activates some kinases, mainly the protein kinase C, which will affect the cell metabolism, proliferation, and other functions required by the cells. Now, this schematic diagrams shows an example of these secondary or second messengers produced by in the G-coupled protein receptors. The third type of receptor is the enzyme-linked hormone receptors. The enzyme-linked hormone receptors, these type of receptors, they have intrinsic enzymatic activity, or these receptors, they can bind closely to another enzyme, which will initiate different intracellular processes inside the cell. Now, an example of these enzyme-linked hormone receptors is the leptin receptors, the insulin and growth hormone, which has a tyrosine kinase receptors, and the natriuretic hormone, which function through guanylate cyclase linked receptors. So these receptors, as I mentioned, they have intrinsic enzymatic activity, or they are associated with directly with other enzymes without the need of intermediate messenger or protein, like in the G-coupled protein receptors. We have also another type of the previous hormone receptor types. They are extracellular receptors. Now we will talk about the intracellular receptors. We have the steroid hormone receptors. The steroid hormone receptors, they are located in the cytoplasm or in the on the nucleus, and the binding of steroid hormones to these receptors will activate the transcription of various genes, which will ultimately be translated into the targeted or required proteins to regulate various processes in the cells. The second type of hormones which acts on intracellular receptors is the thyroid hormones. Thyroid hormones mainly binds to the nuclear receptors, and the binding of thyroid hormones to the nuclear receptors will lead to two things. First of all, it will activate various genes like the steroid hormones, and these genes are required for controlling the metabolic activity on almost all cells in our body. The second effect of binding thyroid hormones to its receptor is that it will increase or control its own functions or its own levels, and this we call amplification of function. So by that, the thyroid hormone function can stay for days or even weeks. Now, this table summarizes the hormones and different types of receptors for these hormones. And in the next lecture, we will start with the hypothalamus pituitary glands. Thank you. Any questions?
دكتورة اليوم حسيت شوي صعب المحاضرة، مش عارفة يعني حاسة كتير
إذا في أي سؤال يعني هلا قديش الوقت إحنا معانا؟ إذا في معانا الوقت اذا في اي اسئله يعني ممكن اجاوب عليها، او اذا بتحبوا مثلا انه تقراوا المحاضره تبعتوا لي اسئله على الايميل برضه بصير.
دكتورة ما بنعرف ايميلك.
الايميلي في اعتقد الدكتوره فاطمه بعثته للسي ار تبعكم.
تمام.
فاذا في اي اسئله او اي بتحبوا اي توضيح يعني عن طريق الايميل، او في عندنا المحاضره تاعت يوم الخميس الفلكسبل لكشر، ممكن نناقش اي شيء بدكم اياه. طبعا انا التكست بوك اللي بستخدمهم لمحاضراتي هم بيرن اند ليفي فيسيولوجي والجايتون.
تمام.
اي
دكتورة
استفسار؟
بس يا ريت لو المحاضرة.
تفضل.
دكتورة كنت بحكي إنه إيش نعتمد ولا المصدر الثاني؟
التنين انا بجيب
أرجع آخر سلايد.
منهم التنين انا بجيب. بس
دكتورة
يعني
بتعتمدي
سلايد؟
نعم السلايدات انا تقريبا يعني من التنين حطيت كل شيء بالسلايدات.
يعني السلايد كافية بالنسبة للامتحان؟
بالنسبه للامتحان
تمام دكتورة.
فيعني اذا
دكتورة
اي سؤال، في اي سؤال بتحبوه مثلا بس بتقرأوا المحاضره انا جاهزه تبعتوا لي اياه على الايميل، او اذا بدكم يوم الخميس بنناقشه.
دكتورة بس حسيت شوي الشرح كان سريع شوي، يعني إذا ممكن بالمرات الجاية يكون أبطأ شوي
تمام.
يعني.
بعتذر ان شاء الله،
ما
خلص
كناش
المره الجايه
نلحق
انا
نفهم.
خلص ان شاء الله انا المره الجايه يعني بكون ان شاء الله ابطأ شوي.
خلاص تمام إن شاء الله.
فان شاء الله يعني اي شيء بدكم اياه بس خبروني وانا جاهزه يعني.
شكرا جزيلا لحضرتك.
بس
الله يسلمك.
بحس انه مش عارفة لو النقطة هاي تاخديها بإيجابية. حسيته كله انجليزي بانجليزي، كله دخل ببعضه في راسي انا صراحة يعني.
خلص يعني احكي المره الجايه قصدك عربي، خلص ان شاء الله يعني اعمل ميكس.
يعني بس انا بدي احاول افهم، يعني مش عارفة دخلوا براسي كل شيء سريع ويعني حاولت.
هي لانه بجوز المقدمه عن الهرمونز والريسبتورز تاعونهم بجوز بعرفش اذا اخذتوا بايوكيمستري قبل هاي المحاضره مش هيك؟
اخذنا.
ايش انا لاني بصراحه البايوكيمستري مش عارفه شو بيعطوا فيها بالنسبه للاندوكراين، هل بيعطوا على الريسبتورز؟ هل بيوضحوا الاشياء اللي انا شرحتها بهاي المحاضره؟
أعطى بعض الأمور عن هاي الأشياء بشكل بسيط، بس يعني لسه ما بدينا بالتفصيل.
اوكي يعني هاي مثلا هاي الموضوع انه طرق كمان في البايوكيمستري صح؟
إلى حد ما نعم دكتورة.
اوكي فخلص اي شيء بدكم اياه يعني خلص المره الجايه اذا هذا بكون مثلا ابطا وبحكي بوضح بحكي بالعربي كمان.
أهم شيء العربي.
خلص ان شاء الله. انا بقى يعني بجوز لاني انا يعني بحس انه احيانا الواحد انه بعض المصطلحات احيانا انه تنحكى بالانجليزي بصير اسهل بالحفظ والهذا، بس مش مشكله خلص المره الجايه ان شاء الله بكون ميكس بين هيك وهيك.
إن شاء الله.
دكتورة إذا بدك أزودك بمحاضرات البيكم مش غلط كمان.
والله يا ريت عشان والله اشوف انا يعني شو بيعطي، لانه المشكله انه الفصل بموضوع بين الفسيولوجي والبايوكيمستري بالاندوكراين سيستم يعني شويه صعب، فبدي اعرف انا شو بيعطي شو بتاخذوا بالبايوكيمستري.
خلاص تمام دكتورة.
اذا ممكن تبعت لي يعني المحاضره اللي اخذتوها اليوم.
تمام إن شاء الله.
ان شاء الله تعالى. اي شيء بدكم اياه؟
يعطيك العافية.
الله يعافيكم.
دكتورة اليوم حتبعتينا السلايدات؟
رح ابعتهم للكوردينيتور الدكتوره فاطمه وهي رح تبعت لكم اياهم. اي شيء ثاني؟
لا يعطيك ألف عافية دكتورة.
الله يعافيكم. اذا موعدنا ان شاء الله بكره بنبلش ان شاء الله بالهايبوثالاموس والبايتيوتري جلاند.
إن شاء الله.
يعطيكم العافيه.
يعطيك العافية دكتورة.
يعافيكم.